Somebody lower the lights, please. Okay, any questions from last time? I'm going to get this back to where we were. Okay, questions from last time? All right, somebody remind us of where we ended last time. What were the 3 or 4 most important points? Give me a column. Just one. So homeostasis, Leonard. Active transport. Crystal. The 3 compartments. Did you have something else to say, Laura?
The endocrine system.
Endocrine system. Anybody else? Come on. Electrochemical gradient. Good. Excellent. All right. So we're going to, if you could, at this point, take out those 3 handouts I talked about. One of them is the homework on osmolarity. If you don't have them, that's fine. It's not the end of the world. One of them is the handout on intracellular and extracellular fluids. And one of them is about penetrating and non-penetrating substances. So I'll need to borrow somebody's. Apparently I gave all of mine out. I was too frugal this semester. All right, let's talk about this first. This one is the intracellular concentrations and extracellular concentrations. Okay, these are the numbers we are going to use. Straight out, you need to memorize this list. Say it to yourself in your sleep, make a song about it, sing it to your girlfriend. It's Valentine's Day. Say, I got a great Valentine's Day song for you. Make a song, whatever it takes. Let me just go over the basics of this, right? Sodium. It's in milliequivalents per liter. So the next time we will do the millimoles to milliequivalents handout. So sodium, probably the most important ion in your body. It is in the highest concentration of any ion. Okay? Very, very important. Sodium is much higher outside the cell. It's about 140 milliequivalents per liter. Now, you don't know what that means necessarily. Maybe you do. Maybe you remember some chemistry. Inside the cell, it's 14 milliequivalents per liter. So it's 10 times higher outside the cell than inside the cell. Potassium is actually reversed. And the extracellular concentration is even lower. All right? So it's even more the difference. Calcium, as a couple of people figured out, is much more concentrated outside the cell. Then we go down to chlorine. Chlorine is also much more concentrated outside the cell. Glucose, outside the cell, very high. Inside the cell, not very high. Okay? Because you're using it or you're storing it as glycogen, so you're not expecting to see a lot of free glucose. Osmolarity. We're going to use a number of about 295 milliosmoles per liter. Again, you don't know what that means yet, but you will. If you look on a bag of saline, a 1 liter bag of saline in the hospital next time you go, because a friend of yours or relative is in the hospital, take a look at the osmolarity. It's roughly 300. Somebody tell me why that bag is roughly 300 milliosmoles per liter. I haven't told you why. Take a guess. Yeah, Teresa.
The higher pressure forces things to remain so that it gets past the pressure rating here.
Nope, but good idea. It's a fantastic idea, but it's not what it is. Anybody ever been hooked up to an IV bag before? Yeah, Mike.
I was going to say that it's the higher concentration, it'll then diffuse into your cells, right?
It's good logic. It's fantastic logic, but it's not the truth. Yeah, Leonard. No, these are all good reasons. Your osmolarity in your body is roughly 300 milliosmoles per liter. So when they give you a bag of saline that's 295, 290, 300 milliosmoles per liter, they're essentially giving you the salt water that matches your internal content. So it doesn't cause fluid shifts. You don't understand that yet, but you're going to understand. They're basically giving you a neutral substance that has all of the nutrients as far as sodium and water that you need. It's neutral. It doesn't do any of that. It's the same as what you are. And you're going to understand as we go through this why it would be maybe not so beneficial for it to be either higher or lower. But it's all good logic, see? So logic can only take you so far. Logic takes you to the next question. But the reality is that this is roughly matches your internal osmolarity. Okay? Is this exact? No. 295, 300, 290. We're going to use 295. pH, super important. The pH outside the cell, roughly 7.4, 7.35 in the blood. A little bit more acidic inside the cell. Okay? So these numbers, I'm not going to pussyfoot around on. Just memorize them. Just memorize them. Okay? You have to know them. And all semester we're going to use them. So it's worth the time and effort. Okay. That. We're going to talk about penetrating substances, okay? Not right now, but this is, well, not very long right now. This is an explanation of what penetrating and non-penetrating substances are. So there are substances, and these are on a scale. We're not going to worry about the partitioning coefficient. We're just going to worry about permeability. These are substances that enter the cell at will. Remember we talked about things that can diffuse simply? These diffuse simply. Now, the rate at which they go in and out of the cell is determined by this permeability coefficient. So, which of these substances most easily gets into the cell? Which of these substances? What is it? No. Methyl alcohol. What's right underneath methyl alcohol? Ah, ethanol. Fantastic. Everybody should be familiar with ethanol. I'm sure, maybe this weekend. So, ethyl alcohol gets into your cells relatively easily. No problem. Easy peasy. Can cause problems with balance, speech, thinking, coordination. Okay? So these are the substances. The other thing, you're going to need to memorize these. Okay? As penetrating substances. Straight up. Not going to lie to you. Tell you, just come out. Memorize. You just need to know the permeability, not the partitioning coefficient. That's a more advanced topic, and I don't want to get into that. So you need to know which ones are penetrating substances and which ones are the most to least. Glycerol is the least penetrating of these. It means that it takes the longest to get into the cell. It does, it's just slow. Okay? Now keep the homework out for now. We'll talk about it at the end of class. All right, let's keep going. So, let's talk about osmosis and osmolarity. Okay? So, and this is diffusion. This all leads into this. So, endo and exocytosis. I guess we ended there. Endo and exocytosis is when the cell opens up. When the cell opens up and you want to get something extremely large into the cell. So we're going to draw the standard cell here. Okay? When a cell makes something, a protein, let's say it's a hormone. This is part of in an endocrine gland somewhere. That protein then gets packaged, right? We've got the whole nucleus and the endoplasmic reticulum and the Golgi apparatus, all of that fun stuff, right? It gets packaged. Let's just say it's estrogen. Is estrogen a large or a small protein? What do you think? It's large. It's a hormone. It's huge. Okay? Now, can it get in and out of the cell? Now, this is a trick question. So I'm going to pick a different substance. Can this get in and out of the cell by itself? It's large.
Steroid.
It's a steroid, so it's cholesterol based, so it actually can. Okay? So that was a trick question. All right, now I'm going to pick something that isn't a steroid. All right? Growth hormone is another hormone. It's very, very large. Okay? It cannot get in and out of the cell by itself. Can it use simple diffusion? No. Can it use active transport? No. Can it use secondary active transport? No. Cannot do it. Too big. Huge. Okay? There's only one way to get this sucker out, and it's polar, and it's a large molecule. So because of that, the cell membrane just doesn't like it. So what has to happen is that this thing has to fuse with the membrane, right? Fuse with the membrane and open up. And the growth hormone is then released. And then this membrane gets reincorporated. That's called exocytosis. It is used to get rid of garbage and to get rid of things that are so large that the cell has to open itself up. All right? Now, is the cell at any point here, the internal environment, exposed to the external environment? No, that's the key, because if it was, it would die. Okay? Endocytosis is exactly the opposite. Let's assume that this is a new cell that's receiving the growth hormone. Right? Same idea, too big. Got to get it in somehow. So it's going to fuse with this thing, and then it's going to allow the growth hormone to come in. That's endocytosis. Everybody with me? Okay, there are two forms of this exocytosis. The first one that I mentioned is called phagocytosis. This is endo. Sorry. Endocytosis. Phagocytosis is this one. The second one is called pinocytosis. Here's the way you differentiate between them. So these are forms of endocytosis. Phagocytosis is the equivalent of a cell eating something. It's big, it's honking. Pinocytosis is the equivalent of a cell drinking. Smaller substances, usually liquid, but still too big to get through a channel or transport. So it's shades of gray here. Right? Shades of gray. Real basic, right? It gets more complicated than this. In your book, they talk about clathrin-coated pits. That's a little bit more complicated. It's something you should know. Note to self. Write it down. Clathrin-coated pits. Write it down. Because Martino said so. Yeah, Leonard.
Endocytosis always polar molecules?
Not always. So Leonard's question is, are they always polar molecules? Because they don't have to be. But they're very large. So, for instance, cholesterol is a hydrophobic molecule. Technically, cholesterol can go through the cell membrane on its own, and some of it does, but there's still what's called this clathrin-coated pit system that allows endocytosis to bring it in. So there's an example of a molecule that just sort of doesn't fit this polar large, right? You'd think it would be able to get in on its own, and it can, but just doesn't always. All right? Because it's okay if it's not all right. But you need to let me know. Okay? Good enough. All right. So, diffusion. Again, some of this is still review, but we're moving through this because everybody I want to be on the same page. So, what this is showing here is what? What do we have here? What is this first diagram A showing us? Somebody tell me what that is. So I like to refer to this as over time, the solute molecule is replaced. The solvent is the liquid that it's in. The solute is stuff. I try to simplify things as much as I can. It's stuff, right? So what does this diagram show about this stuff, Laura?
Stuff is more concentrated on one side than it is on the other.
Yeah, on the left side and the right side. So there's a what? I'm very technical here. What I asked you to do again. The electrochemical gradient favors moving from the left to the right. Yeah, there's a gradient. It's probably no electrical component. Let's say that it's electrically neutral. So what would eventually happen is diffusional equilibrium. So far, so good? Okay, this is another experiment. If you've taken Professor Greening's Bio 1040, she actually does this. It's a simple diffusional experiment with compartments. You stick a bunch of salt on this side. You have a semi-permeable membrane, which mimics your cell membrane, right? So semi-permeable means it only allows certain things to get through. Okay, in this case, it's salt. You start a timer in A. There's a electrochemical gradient on the left in 1. By time B, there's stuff moving in this direction and in the other direction, right? It's called Brownian motion, random motion. Stuff goes in both directions, even though there's a concentration gradient. And by the time we get to C, there's an equal distribution. When they are in equilibrium, does that mean that there are no stuff, molecules moving back and forth? No. But if you sample at any given time, the likelihood of you finding an unequal amount of stuff on either side is pretty unlikely, even though things are constantly moving. That's all I know. Okay? And so this is simple diffusion. Now, there's a plateau here from compartments. In this case, they're using glucose. If this was true simple diffusion, would you see a plateau? There's something very, very interesting about true simple diffusion. It's very, very important. You know this isn't, because I told you it's not. How does glucose get into a cell? What does it need? A transporter. Is that simple diffusion? That's called what? Facilitated. You have a transporter. It's facilitated diffusion, which means that you're eventually going to get a plateau because why?
It's going to take equilibrium at some point.
Well, the equilibrium, but that's not this. This is a bigger issue than the equilibrium. Yeah.
Or you won't be able to set up a gradient for the secondary and transport.
No, it's simpler than that. It's big picture. See, Teresa.
The rate at which the glucose can be transferred into the cell is fixed.
It's fixed because you can only get so many transporters into the cell membrane, and you can only get the transporters to move so fast. In true simple diffusion, is that a problem? So in true simple diffusion, this should be a perfectly straight line at 45 degrees or a perfectly straight line at 45 degrees. Whenever you see something like that, there's a limiting factor. Oxygen doesn't have a limitation. Oxygen can go back and forth between your cell at will. The more oxygen, the faster the diffusion. When you see this, there has to be a limiting factor. In this case, it's the number of transporters and the speed. Does that make sense to everybody? So this can't be simple diffusion. But it is diffusion because it does run an electrical chemical gradient. So far, so good? No.
So if you have more transporters available in this membrane, this graph would not be the same.
Correct. If you had more transporters available, you wouldn't. But would you ultimately come to a plateau even when you added more? Right. Ultimately, you're still limited by the number of transporters. And that's so maybe you can extend this, but there's a limit to how far you can extend the transporter. Okay. So this is called flux. There's actually an equation for this. You don't have to worry about the net flux equation. All right. So this is one-way flux, and this is vectors, right? If you were doing physics, you'd do this in vectors, but we're not doing physics. All right. So most of the flux wants to go in this direction. A little bit of the flux wants to go in this direction, just due to random motion, and ultimately there's a net flux. Does that make sense? Now, if this is not a semi-permeable membrane, if any of you have already done that first Physio X lab, if this is not semi-permeable, what would happen here? Let's say that this molecule here, it's not semi-permeable to this molecule. Max.
Build up pressure on that side.
Yeah, you build up in pressure. And that's what's called an osmotic substance, a non-penetrating substance. Okay? We're going to start talking about non-penetrating substances. Now, most ions are non-penetrating substances. Okay? So, if the membrane setup is not permeable to the ion directly, then the ion will stay on that side, in this case the left, and pressure will build up, and that's called osmotic pressure. Okay, these are what ion channels look like. So in order for sodium to move, we're gonna focus on sodium, because sodium, think about it, when you become dehydrated, what are the three most important things that you need when you're dehydrated? Salt, which is usually, there's lots of electrolytes, but the most important one is sodium and then glucose. Okay? Those are not easily permeable. Those salt and the sugar can't easily get into and out of the cell. They need help. Okay? The salt and the electrolytes, because there's lots of them, use ion channels. Are those ion channels always open? No, that's the problem. And there's a certain number of them. You can't make an ion channel run faster, because all an ion channel is a tube. That's it. And there's different states, right? There's open, closed, and resting state, where it's not really open or closed. So if you were in, I think Dr. Burling's actually teaching structural biology this semester, right? And so he actually looks at this in this format, and they actually look, it's all computer, it's really, really cool course, it's going to be around in a couple of years. But he actually looks at channels, and it's called X-ray crystallography, and he takes all of these apart, which is pretty cool. But I don't care about X-ray crystallography. For me, this is what it looks like, it's a nice picture, but it's not really necessary for this class. Okay. That sheet that has the intracellular and extracellular concentrations of those things on that sheet, what are the majority of those substances on that sheet called? They're called what? Ions. Okay, then there's glucose and pH. I think I took out bicarbonate. No, bicarbonate's in there too. Okay. All right. So here's where we get into, why do I say electrochemical gradient? Why am I such a stickler about that? Well, one, it's because thermodynamically speaking, it's correct. But two, ions and the distribution of ion changes the charge inside and outside the cell. Why is this important? Because when we get to the neuro and muscular portion of physiology, you have to understand voltage and current. It's not because it's chemistry, it's not because it's physics, because it's physiology. So there's an unequal distribution of ions inside and outside the cell. How do you know that?
Proteins are usually negative.
Right. But there's an even easier, that's true. Proteins are usually negative. Because I gave you the sheet that says that there's an unequal distribution on it. But he's right, absolutely. That sheet shows you the unequal distribution. And it's because of those numbers that you have a difference in charge inside and outside the cell. Generally speaking, the inside of the cell usually is more what? Negative or positive? Correct. The inside of the cell, generally speaking, is more negative than the outside of the cell. So when we get to muscle and neurons, we'll talk about the resting membrane potential being negative 60 millivolts, negative 75 millivolts, negative 90 millivolts. So when you're talking about a concentration gradient for an ion, are you only talking about the concentration gradient? No. Somebody other than Leonard, I know he knows the answer. Why are we not just talking about the concentration gradient? Why is that ion do more? Max.
The difference in charges and great voltage.
Yeah, ions carry charges with them. Their movement carries charges. And so they not only have a concentration gradient effect, they have an electrical current effect too. It makes a difference. You're an electrochemical being. You're a combination of chemicals and electricity fused together. You're like a giant battery, is what you are, essentially. Okay? You don't think of yourself that way, but that's what you are. So the distribution and knowing the distribution of the ions, that's what this shows you, right? Does this mean, so here's a point of confusion. What does this picture show on the outside of the cell? Positive charges. Does that mean that there are only positive charges on it? No, it's a theoretical construct, meaning that the majority of the cells on the outside, the charge is positive. Don't get confused. Say, well, you showed me that diagram and there were no negatives on the outside of that cell. Well, of course there were. Leonard just said it. Proteins carry negative charges somewhere. Okay? Does that mean that there are no positive ions on the inside of the cell? Well, you know there are because potassium has a +1 charge on it. And it's sitting mostly on the inside of the cell. Okay? So this, if you took all of the charges of all of the ions inside and outside the cell, there would be more positives on the outside and fewer on the inside, and that means that this is more positive and this is more negative. And that's what that's saying. Is everybody okay with that? I'm throwing some stuff at you. You may have heard it before, you may have not heard it before, so you may be reviewed. But I need to clear things up. If you're not knowing what I'm saying, if you're not understanding, I need to know. Because these are key concepts. Remember, what does it say on that sheet in the syllabus? It says basics of physiology. This is the core. You can't say, I'm going to do this next week. You can't say, I'm going to understand that tomorrow. There is no tomorrow in this class. It's all about today. Okay? That's what you, so when and if, don't be embarrassed, because if you don't get it, I bet you 5 or 6 people in each class don't get it. Yep.
So with the diagram, it shows positive going in and negative going out. Does the cell want to, inside of this, does it want to get rid of negative charge, or does it want to bring in more positive charge? Again, that's kind of like the same thing.
It is. But it's, see what Leonard's asking? Yes. So opposites attract, right? If the outside of the cell is mostly positive, then where do those positive charges want to go? In. And if the inside of the cell is mostly negative, where does that want to go? Out. Generally speaking, that's correct. Now, what prevents that? The semi-permeable membrane.
Does it form a cycle where positive goes into the cell, but then goes negative out, and then you have more negative outside?
Yes, it does form a cycle. And the cycle is because channels and transporters open and close. But what is constant? We talked about active transport, primary active transport last week. And what was the example that I used for primary active transport? Sodium, potassium, ATPase. It is ubiquitous. That means it is everywhere. It is that pump system that creates this gradient. That pump, if it shuts down, destroys this gradient. If this gradient doesn't exist, you die. It's as simple as that. It's the pump that generated the gradient and also generates a charge difference, a really small one, 3 millivolts, but it generates a charge difference. So you're exactly right. It is a cycle, and then when things open and close, it's the job of the pump to keep the gradient as is. Because what do you use sodium for a lot of? What is its primary goal? I mean, it has lots of goals, but it has a goal. Why do I want to keep, yes?
Is that like glucose?
It does. Yes, but what, more general than that.
Water.
Say that again?
Water.
Water, yeah. Even more general than that. If I'm thinking about sodium, why do I care so much that the sodium outside the cell is so gosh darn high? And I want to keep it that way. Charge and
For
secondary active transport. It's an engine. It's potential energy. See, we're getting more to the basics of chemistry and physics now. That sodium is a huge potential energy reservoir that does so many different things. And if that reservoir goes away, your ability to function goes downhill very, very quickly. That's why it is so important that there are charge differences and your cells have a semi-permeable membrane. It's potential energy that drives secondary active transport. And yes, you're right, it brings in glucose. And yes, you're right, it can bring in water. But without it, you can't do any of that. Super important. A lot of really basic concepts. You know what I'm doing? Do you see what's happening here? I ask a basic question, a global question, and I get real specific answers. What I'm trying to do is bring you back first. Do you understand the major concepts? And then we'll get into the nitty-gritty of the details as we go along. But if you don't understand this big concept, I'm looking at it from a 30,000-foot view, and you're already under with a microscope. Too soon. Okay? So try to think broadly and then come back. So you're at telescope level, satellite level, and then you're coming back to the microscope, and we're going to go back and forth all semester long. And you have to be able to distinguish the difference. Okay? It's a lot of information. All right. Questions? Hear that? I'm just clearly one here. Okay. So these are open channels and closed channels. Some of this stuff is really, really basic. Okay, so don't worry about this. This is just what happens with a transporter. They draw a transporter like this, right? In the case of the glucose transporter, glucose binds to the GLUT4 or the GLUT1 transporter, and it gets moved in the direction of the electrochemical gradient. Oh, very important graph. This goes back to what I was saying. And what I was saying is that true simple diffusion has a 45-degree angle. That means the more stuff that you have that uses simple diffusion to move, the higher the rate of transport. And there's no limits. It is limitless. In theory, yeah.
But aren't you still limited by the number of transporters?
Oxygen doesn't need a transport. CO2 doesn't need it. Gases don't need transporters. There's no limits. Yes. If the substance, now, this is mediated transport. So it plateaus, just like it did before with the group transport. Now, what if I had a third curve that did this? What would that show? Now you got to go back to some like rad, sig, world, device, and wherever you're at. Okay, the third curve starts off just like this and does this. It goes right below and hangs right below mediated transport. What would that imply?
The rate of reaction slow.
Okay, well, or competition. You have a competitive inhibitor with me. Right? Competitive inhibitor. So by looking at these graphs, you should be able to see things like that. Again, it may be complicated, but this is very, very basic. You have to understand these basic concepts because they start to build very, very quickly. Okay? Is everybody with me? Don't look scared. Right? So, in a simple diffusion, flux rate is limited only by the concentration gradient. The more of it you push in there, the faster you can get it to go. When we get to the respiratory system, we're going to talk about when you use pure oxygen in a clinical setting. And you have to understand how oxygen moves and why. Why it's a good idea, why it's a bad idea. And a lot of times it's a bad idea. In carrier-mediated, carrier exchanger, whatever you want to call it, transporter, the number of available carriers places an upper limit on the flux rate. All right, so these are just examples of a semi-permeable membrane, right? This is simple diffusion, this is facilitated diffusion, this is active transport. We've already talked about this. This is just the visual. Okay? These are just different types of transporters. These are just visuals. I've already talked about this. This is for you, not for me. Okay, now, remember what I said about that sheet? I said, wait until we start getting into this. Somebody's going to say, well, Dr. Martino, they say it's 150 millimolar. And your sheet says it's 140. Yeah, I heard the course. It's 140 because that's what I say it is, because that's the easiest way for us to all be on the same page. Okay? Is that the exact number? Is there an exact number? Of course not. I'm trying to make it easier for you, not to confuse you. Right? I'm trying to simplify things, believe it or not. That may seem counterintuitive, but that's what I'm trying to do. And so these are the different things that happen, right? So we've already talked about this pump, and now they're starting to bring in these concentrations, which are a little bit different than yours. And I have it in milli equivalents, and they have it in millimolar. I'm going to show you how to go from one to the other. You're going to learn how to do that calculation if you forgot how to do it from a chem. Or maybe you never learned. But I'm going to show you how to do it. Okay. Again, don't worry about any of this. This is just real basic. Okay? You need to know about co and counter transport. Again, real basic topics. So, in the case of, you know that outside the cell there's a high amount of sodium, and we showed an example that was like this. Actually, it was like this, right? The sodium-calcium exchanger uses the potential energy of sodium to pump calcium out of the cell. You can do the same thing with something that is low outside the cell and wants to go to high. You use the potential energy of sodium to do that. So this is called co-transport. They're going in the same direction. Okay, there's other names for this stuff. And in cell molecules, different terminology.
So that's facilitated diffusion, right?
This is a type of facilitated diffusion. Correct. But this is also a secondary active transport system because this gradient was set up by what? What set that up?
Sodium, potassium.
The sodium, potassium, ATPase set that up. So this is also secondary active transport. Secondary active transport is a type of facilitated diffusion.
So secondary active transport doesn't use ATP.
Directly. Indirectly. ATP created the gradient that you're using. Does it have to be sodium? No. Are all secondary active transport systems based on sodium? No. Why do I use it? Because most of them are. And it's the easiest version to deal with. You need to memorize all these transporters. These are primary active transporters, secondary, facilitated. These are examples. They're not all of them. Ion channels, but these are all ion channels. There's magnesium channels. There's all kinds of channels. Right? So this is straight out just memorized. See, this is one of the examples I was telling you about. This is a chloride bicarbonate transporter. This is really important in your kidneys and your lungs. So this is really advanced. Here, most of them use sodium. Yeah, Laura.
If I memorize, we just have to know what they do, or do we have to know, like...
I want to know examples of all of them. So yeah, you have to know what they do. Well, what they do is it's self-explanatory, right? But yeah, you have to memorize them. Because I may ask you a question, right? And that question is based on, oh, I assume that they understand this. I may not ask you, give me 3 primary active transport systems. That's too easy. I don't want to insult your intelligence. That's when you were a freshman. You're not freshmen anymore. Okay? So yeah, so that assumes, it's implied that if you know what they are, that you know what they do. They just move stuff in the direction that it says they move. Yeah, Leonard.
So I don't know how glucose can be part of the secondary active transport.
Glucose is not part of secondary active transport in this case because we're not using it in that sense. Is there some? Are there some? Absolutely. There's the sodium glucose transporters that use the sodium gradient. But we're just trying to, this is for when you're going from the blood into the cell, the simplest format. But is there one? Sure. Are these all of them ones that exist? No. There's lots of them. We're just giving you a few. Yeah. Crystal.
Just like handout.
An example of
Like anything like that. We have to memorize it.
a type of a question?
Like, where would you find the chlorine ion channel? Or where would you find
Oh, the chloride bicarbonate
yeah,
channel?
Can you give an example?
In the lungs and so in the nephron, so this is why I'm taking so long, because I'm going to start confusing the hell out of people right now. All right, so when we get to the kidney, right, the kidney, the functional unit of the kidney is called a nephron. All right, and within the tubules, you have movements of ions like bicarbonate, which is a buffer, right? The major buffer in your system is bicarbonate, and chlorine and bicarbonate exchange, right? They exchange, and the reason they exchange is because you may become acidic, and when you become acidic, your pH drops. You need to get bicarbonate into the system. And when you become basic, you need to dump bicarbonate out of the system. And one of the ways to do that is to counter-transport with chloride. Okay? There's something called carbonic anhydrase, which is an enzyme involved in this whole chemical reaction. You see how I'm starting to tread lightly here? Because I'm going in an area that you're not ready for yet. So, I would ask you a question, for instance. Why, let's say I give you a bogus ion X, okay? And I told you that ion X has a electrochemical gradient that favors it moving outside of the cell, and I want it to move into the cell, okay? How could you envision connecting ion X to a transporter to make that happen? Have I asked you to give me any of these? No. What I'm asking you to do is understand the concept, and the concept is what? What would you have to connect ion X to get it to move against its concentration gradient?
Secondary attitude.
And which one? It depends on which direction you're going. Right? So it could be potassium or sodium. Is that asking you directly one of these? No. Is it harder? Sure. Because you first have to understand how this works, and then you have to say, how can I link them up? That's an easier question. Yeah,
Is there a way to tell if something's secondary active transport or primary active transport, or is that just something you have to memorize?
is there a way to tell?
Based on like the two ions that interact with each other, or is there no one particular path?
Look at. You have to. Yes, you can if you have a transporter that only moves one molecule, and you know all of that sheet, the intracellular and extracellular concentrations, and you know that the ion favors a gradient, an electrochemical gradient that goes into the cell. You know it because I gave you that sheet. And the thing is moving out of the cell. What's the assumption you have to make? And it's not connected to anything. So you do some of the molecular biology, and you know for certain that there's no other ion connected to this channel. Then what's the assumption you have to make? You know that the gradient favors it moving from outside the cell to the inside of the cell. It's moving from the inside out. And you know that you've done some biochemical molecular test to search for other ions that are associated with this transport. You can't find them. All this thing is doing is moving this ion Z out of the cell. But you know that ion Z has a gradient that favors it going into the cell. The assumption has to be what? It has to be. You could test for ATP usage, right? You could do the biochemistry and see, oh, there's cranking around here. There's a lot of ATP being used in this area. So there's lots of tests you could do. But in the simplest answers, just memorize the ones that are. But if I ask you a theoretical question, that's how you have to think about it. Do you see what I mean? You have to understand the concept. Okay, how do these things work? And then if he gives you some mumbo-jumbo planet Z in universe Q question, oh, what the hell's planet Z in universe Q? Does that even exist? Then you have to understand how the system works. So now you can't just memorize it anymore. Now you have to understand conceptually what it does. Make sense? You can all do this. I know there's a lot of scared looks. You can do this. Trust me. I've been doing this a long time. You're not any different than any group that's ever come before you. So you have to trust them. I think we're done for now. So the homework we'll deal with at the beginning of the next class.
Do you want us to do it?
No, you're not ready for it. I wanted to talk about it. No. Yeah. Don't even try. Talk about panic attacks. Okay, three most important things. We didn't go over as much as I wanted to, but we went over a ton of depth. Right? There's a lot of meat to what I'm talking about here. Three important things. You can give me one. You don't have to give me all three. Alexis.
The difference between intracellular and extracellular.
Have to know it. Stuff that's memorized. The stuff that I said to memorize, you've got to memorize today. Do not wait until tomorrow. It is too late. Don't do that. Don't make that mistake. Yeah.
Do you want us to do like exactly that? Like 3 sodium, 2 potassium, or just
Yes. For the sodium, potassium, ATPase, you have to know 3 and 2 because that's electrogenic. That's what makes it electrogenic. Yep, that one is specific. Crystal.
That there's not just positive and negative.
Yes, that there's not just positive and negatives outside. Yeah.
Like understanding where the fusion plateaus.
Yes. If you see diffusion plateau, what is it? Think of the big concept. So there's 2 things I'm asking you to do today. There's big picture stuff, and then there's minutia detail. And I've tried to separate them out. The stuff that's minutia detail, just memorize it. Don't think, just memorize. The stuff that's big picture, think, and don't memorize. If that makes any sense. That's what I'm asking you to do, all semester long. You'll get used to it. All right. Have a fantastic day.
I have the sheet that I need.
Off because it's still recording.